So in this video, we're going to think about some of the principles of biological signalling pathways. So cells are responding to all sorts of signals from their environment all the time. So it might be stuff from the external environment, like a pheromone transmitted from male to female, or it might be a signal in the internal environment of a cell. So it could be something like insulin coming to regulate blood sugar. It could be something happening at the synapse. So cells are constantly exposed to signals. And what cells need to do is to turn those signals into appropriate biological responses. So they need to be able to control what they're doing. So in this video, we're going to think about some of the architecture of signalling pathways. So there are plenty of other videos I've made of specific signalling pathways. This one's going to give an overview of the sorts of principles of signalling, the overall design plans, to give you an idea of how signalling pathways are organised. Okay. So obviously, if we're thinking about a pathway, we need to start with a signal. So as I say, that could be a small molecule, it could be a protein, it could be something abiotic, so which could be light, for example. That obviously needs to be perceived by some sort of receptor. So we need something in the cell to be able to detect the signal. That's usually a protein, but it doesn't have to be. So we've got a signal binds to a receptor, and then biologically we're going to have some sort of target protein that is going to cause a response. So target proteins could be they could be enzymes, they could be ion channels or transporters. They could be transcription factors if we're trying to switch on a gene. It could be other things within the cell, but those are the three most common target proteins that we have. So if we activate the target, we'll get a response. And between the receptor and the target, we have a number of steps. So sometimes it might be just 1 or 2 steps, or it might be about 20 steps. So we have a number of steps in there, which I'm just going to summarise by arrows. So that would be the cascade or the pathway. Okay, so we've got a series of different events that happen to connect the receptor to the target, and therefore we've given the response. So that's conceptually what we're talking about with a signalling pathway. But often it's a little bit more complicated than that, in that we don't just have linear pathways, we can have whole signalling networks. So it might be, for example, that the receptor activates these processes, but at this point we have a protein that activates multiple different proteins. So we might have a second target protein that gives a second response. Okay, so conceptually, that split there we would call signal divergence. Okay, so rather than just a linear pathway, the pathway splits. So we have 2 targets that we're focusing on. But it might get more complicated than that. So we might have a second signal that has its own receptor, that has its own cascade, but it might also end up targeting this target 2. Okay, so here, where we've got 2 different signals combine and target onto response, here we just call that signal convergence. Okay, so a second signal has come, but it's actually affecting the same target protein. So we've got 2 different pathways. Sometimes with convergence, it's an AND system, so you need both of them to regulate it. So it only gets switched on if you've got signal 1 and signal 2, or it might be an OR system, so you can have either of them. Okay, and some pathways you need both, some pathways it's either. We can also have interactions between pathways. So we might have a system here whereby the signalling system from signal 2 is affecting the pathway for signal 1, so that will be an example of crosstalk. So you've got pathways sort of interacting with each other. And then the other thing that you often have in signalling pathways are feedback loops. So let's say, again, with receptor 2, activates a few proteins, and then maybe, as a result of that, there's another pathway that comes back to inhibit the original receptor. So that would be an example of feedback. In this case, it's negative feedback. You can also have positive feedback. So the pathway is responsible for its own downregulation. So it activates something, but then maybe on a slightly slower pathway, the pathway is responsible for switching itself off again. So that would be a negative feedback system. So when you see signalling pathways in textbooks, the diagrams often look quite overwhelmingly complicated of all the different proteins. But one of the things you should do when you're looking at signalling pathways is to try and think about it in these terms. Forget about the individual proteins. What are the processes going on? Is there any divergence? Is there convergence? Is there crosstalk? Is there feedback in there? So if you can think about it conceptually before you think about all the individual proteins, then that will help you understand the pathway. So the next thing we need to think about is transduction of information, because each of these steps is one protein activating another protein, or sometimes it's non-protein as well. And there are two main mechanisms by which one protein can activate another. So one is through what we call conformational change. So this is all about shape changes. So let's say we've got a receptor that has this sort of shape, so that will be in the inactive form. When that receptor binds its ligand, binds the signal, it changes its shape. So the whole of the rest of the protein has changed, or it might be just a bit of the protein, but the protein has changed its shape. And as a result of that shape change, it can now bind to something new. So here's our next protein. So it couldn't bind this square form, but it can bind that more rounded form. And then that can go away and carry on the pathway. Okay, so we've activated it by changing the shape. That means another protein can come in and bind, and then that stimulates the pathway. And conformational changes are reversible. So if you stop binding the signal, then you go back to your original shape. The other major strategy we have is covalent modification. So the first one, it was just the shape change of the protein, but chemically it was just doing the same. It was the same thing, but it changed its shape. With covalent modification, we actually change the chemistry of the target a little bit. So here we would have a protein which is inactive, and what we can do is to add things to that protein. So in this case, and by far and away the most common strategy, is to add a phosphate group. And again, that phosphate group then provides a new binding site. So we might have a protein that binds like this, which again will take down the information. So in the inactive form, without the phosphate group, it can't bind, but in the active form, it can. And that gives us two really important enzymes that are involved in covalent modification. So adding a phosphate group to another protein is something that is done by a kinase. So kinases add phosphate groups to other proteins. Okay. And this is reversible, so we can go back. And the enzyme that does that is a phosphatase. Okay, so kinases add phosphate groups, phosphatases remove them. So usually adding a phosphate group activates it, but sometimes phosphorylation inactivates. So in that case, you might have an inhibitory phosphate. So let's say you've got a phosphate in the active site of a protein. That phosphate is inhibiting, and it would actually be removing that phosphate by the action of a phosphatase that would activate your pathway. So it can be that the inactive form is the one with the phosphate group. It can happen in both ways. Okay. So we've now got conceptually what's going on with our pathway, and we know how one protein could activate another. So let's now think about pathway components. Okay, and I'm going to do this with a kind of stylized signalling pathway. So I'm not going to talk about a specific signalling pathway. This is kind of a representation of a typical sort of a pathway. So I'm not going to populate with these with actual proteins. Okay, I'm going to focus on the concepts. So let's think about our cell. And for the sake of this illustration, I'm going to make this one an example of a protein where its target would be something in the nucleus. So it might be a transcription factor, for example. Okay, so we've got the outside of the cell, we've got the cytosol, and we've got the nucleus. Okay, so conceptually, what are the sorts of things that are going to be present? Okay, so let's start. We've got a signal on the outside. Okay. So one of the things that you might have is obviously a receptor. So if you've got a signal that is on the outside, and that always stays on the outside, it never goes inside the cell, you're going to need some sort of transmembrane receptor. So for signals that can't get into the cell, you need some sort of transmembrane domain receptor. So you've got an external bit and an internal bit. Mm-hmm. But we can also have intracellular receptors. So we can also have, so here's a receptor here. Okay, so this is for a second signal. So this is a signal that can diffuse in. Okay, so this is a second signal that is able to get inside the cell. So if it can get inside the cell, then we have an intracellular receptor. So not all receptors are at the plasma membrane. Okay, so let's call this protein A. That would then go and activate protein B through either of these mechanisms, conformational change or covalent modification. We then might have another protein C. So we start to build up a cascade, and notice that in this case a lot of it is happening at the plasma membrane. We have a number of membrane bound proteins. So if it's a transmembrane receptor, we tend to have the first few stages are at the membrane. Obviously, if the receptor is already in the cytosol, this would be less important, but a lot of pathways have a lot of membrane-bound proteins. So we've got three proteins here. So we've got a receptor, activates B, activates C, but not everything in a signaling pathway is a protein. So what you might have is enzyme C is able to catalyse the production of what we call secondary messengers. So these are small non-protein intermediates. So not everything has to be a protein. These are little molecules. So you might have come across cyclic AMP or calcium or IP3. These are not proteins, but they're made in signalling pathways and are equally able to carry the message on. Okay, so C in this case is an enzyme that is catalysing the production of one of these secondary messengers. Okay. Then the next thing we could have in the pathway, we could have, so let's have another protein, protein D, that then binds to one of these secondary messengers in there to become active. And then let's imagine that protein D is a kinase. So we might have that protein stimulates the regulation of other proteins. So let's have protein E gets phosphorylated by protein D. So we put a phosphate group on it. And then we might have a second round of phosphorylation. So we might have protein F gets phosphorylated by protein E, for example. Okay, so what we're building up here is something that's found in a lot of pathways, would be something that we refer to as a kinase cascade. So rather than just having a single kinase, we have multiple kinases working together. So E is a kinase, so it gets phosphorylated by D. D is a kinase, it phosphorylates E. E is a kinase, it phosphorylates F. Okay, so we've got multiple levels of kinases, and that gives us the really important property of amplification, which is the process of going from a weak signal to a strong signal. So if each kinase can phosphorylate 100 target proteins, that means that if you've got kinase cascade, so one phosphorylates 100, and then each of those 100 phosphorylates another 100, you end up going from a very strong weak signal to a strong signal. Secondary messengers are also a point of amplification. So here we might have had only one copy of protein C got activated, but it made 5,000 molecules of secondary messengers. So that's also a point of amplification. Okay. So where are we going to? We might have protein F translocates into the nucleus, so we can have subcellular localization being important. Not in all pathways, but in some pathways where the protein is important, so that's a step that happens. And then we might have our final protein then goes and activates the target. So let's say, so T is my target protein, that gets phosphorylated. So F would be a kinase again, and then that would give us the response. So inside the nucleus, that's likely to be gene activation, but it could be something else. Okay, so T is the final target protein. So in this case, it's something in the nucleus, probably a transcription factor, but I'm trying to keep it quite general. So T is the target, that gives us the response. So we've had a series of different proteins. We've got A was the receptor, we've got some intermediates that are doing either conformational change or covalent modification. We've got an enzyme that made a secondary messenger, so a non-protein component of the pathway. We then had a kinase cascade, so D phosphorylated E, E phosphorylated F, F phosphorylates T, so we've got amplification in that way. We've got subcellular localization, and then we've got the response. But we can also remember we can have crosstalk between our pathways. So let's say in this pathway, here's this receptor protein, let's call it Z. So what Z might do is to go and interact with protein D, for example. So it might inhibit it. So that's where we start to get crosstalk. So it might be that if this second signal is present, then Z also interacts with another protein. So this protein is switched on by a secondary messenger, switched off by protein Z. So that's where we start to get biologically these sorts of crosstalk and feedback mechanisms. So signaling pathway diagrams can look pretty overwhelming of dozens and dozens of proteins inside the cell. What's important is to think about the design of it overall. What's a receptor? What's a secondary messenger? Are there any kinases in there? Are we amplifying? Are we having crosstalk? Are we having conformational change? Are we having covalent modification? So that's much, much more important for understanding this topic than knowing all the names of the individual proteins. So think about the processes and the design before you get bogged down in all the details of all the proteins.